So dan make also try to share screen and see if everything is okay. Hi, Kavini. Are you there?
Did we choose that, what is that password? Did we?
Hello, good afternoon, Somdata.
Hi.
Hey, I've sent a reminder message to all the teachers. I think they'll be joining in 5 minutes or so.
Sure. Okay.
And yeah, obviously we'll be having a little less strength for the session today because all children are at home and they're having online classes as of now. And I'm not sure if they would have data or internet available.
Sure. Yeah. In the meantime, Pavani, shall we try sharing the screen?
Yeah, yes. I request Yasholata ma'am, who's my colleague. She is hosting the session.
Okay. Currently, we are not able to share screen. It says host has disabled that. So,
yeah, Yasholata ma'am, could you please make Somdatta ma'am or Dr. GSN Reddy?
I haven't logged in yet. I'm still in the same session as Dr. GSN Reddy.
Yeah. So please,
make Dr. GSN Reddy himself as host. I'm going to sit here with him.
Yes, that would be great.
Yeah. And I will also log in later on to do the quiz. But right now, I'm not going to do that. Otherwise, with two devices in the same room, it will be very noisy.
Reddy sir is the co-host now. Could you please try sharing the screen now?
Yeah, works. Looks okay? Can someone confirm if you can see the screen?
Yeah, we can see the screen, Somdata. But you need to just switch to full screen view before the presentation. But we can see the screen, it's visible.
Do you see full screen now?
No.
Maybe in a couple of seconds, hopefully.
The screen is visible.
Do you see the full screen now?
Yeah, slideshow.
Great. Let me now try changing slides. Okay.
Up and down.
Okay. Screen also moves, right?
Yeshoman, could you please mute everyone? Make me the co-host if possible. Please mute all the participants. I request the participants also to check their devices and see if they're on mute mode. Thank you.
Pavani, the screen's moved, right?
I can't see it. I can see still the slides. I mean,
Screen is visible. PPT, what do you want to become?
I request all the school teachers to ask their students to join because we'll be having a very fun quiz session at the end of the lecture today. Dr. Somdatta will be hosting the quiz. It would be really fun and interactive if the students could join, because we sort of prepared the questions keeping the students in mind. So we'll start the session sharp at 3 o'clock. Thank you.
Vinny, do you see the screen saying exploring extremophiles now?
Good afternoon, ma'am. Good afternoon, ma'am.
Good afternoon.
Padma ma'am, it was very less. 16.
Yeah, hello. Good afternoon, everyone.
Good afternoon.
I think most of the most of the
afternoon.
students have joined. Yes, yes. Thank you everyone for joining on time. So our speaker for the day, Dr. GSN Reddy, has also joined. So let us start the session, okay? Are you all interested to learn more about microbes, students? Have you all watched the video?
Yes,
Yes, ma'am.
Yes,
Yeah,
madam. We have so much interest.
nice, nice. So sir will be talking more about the microbes today, okay?
Okay, madam.
So let me introduce Dr. Gundlapalli Satyanarayan Reddy. Sir, he is the speaker for today's session. He is a principal technical officer at Center for Cellular and Molecular Biology, Hyderabad. And he has completed his PhD in the area of bacterial diversity from CCMB. And sir has completed his postdoctoral studies. That means that you do the research that you do after your PhD from School of Life Sciences, Arizona State University, which is in the US. Okay. And he is a recipient of many awards and scholarships and fellowships. He received the prestigious UNESCO Merson's SGM Short Term Fellowship in the year 2002. And he has also participated in the iron fertilization experiment, the LOHAFEX. You might have seen, right, where in the video where the scientists have traveled all the way to Antarctica to collect bacterial samples. So sir was also in that team that traveled to Antarctica to collect the samples. Okay. And then he has published more than 69 research papers in peer reviewed papers, journals, and he more than 5 review articles and also book chapters in Burgess Manual, which is a very popular kind of book in bacterial systematics. Okay. And then he has also conducted several skill development programs at CCMB in the area of biodiversity and molecular systematics. And he's also invited at IMTech Chandigarh, NIEST at Jorhat, and CLRI at Chennai for conducting training programs for the students and faculty. Dr. Reddy sir is a member of the Society of Biological Chemists of India, member of the advisory board for the Department of Biotechnology, Gulbarga University, Karnataka, and he's also a senior editorial board member of the American Journal of Current Microbiology. Okay, so it's and as a part of academics and research, he has visited several countries like Germany, Mexico, South Africa, Chile, and USA. So it's really a privilege for us to have such an eminent scientist from CCMB to join and address us and teach us more about the microbes and the kind of study to do that they do there in his lab. So sir, it's a honor to have you, sir, with us, and we are looking forward to learn more from you, sir, during the session. On
Thank you,
behalf
Dr.
of Team
Pawni.
Berries, I welcome you. We extend a very warm welcome.
Thank you, Dr. Pawni, for the nice introduction.
It's over to you, sir. Now you can start the presentation, and later, once you complete the presentation, we'll keep the session open for the students to interact with you if they have any questions.
Sure.
Thank you, sir.
What do you want to become?
Welcome to the session. All the children, can you hear me?
Yes, sir.
Yes, sir. Yes, sir.
Yeah. Can you see my slides?
Sir.
Okay. Nice. Thank you. So, welcome again. I'm thankful to Dr. Pawni for the introduction. Actually, it would have been very nice if there was a physical interaction between me and the student. But nevertheless, I think we'll try our best to have that kind of interaction through online. Okay? So, students, what do you want to become in your life?
Doctor.
The problem is like,
Yes. Doctor, sir.
Doctor. Yeah, so, okay. If you want to become a doctor, only sick people will come to you, right? Or you can make money when people are sick. Because the society has to be sick, you're good, right? Correct. Want to become a lawyer, and there has to be some kind of crime in the society, and only then you can prosper, right? So I don't think you would like to become a lawyer or a doctor that way. And in engineering, once you make a house, you need engineering services. And a politician now, what kind of politicians we have, right? I don't think anybody would become a politician that way. So, what do you want to become? The moment you get up from bed, you go to take your brush, right? The paste, correct? Okay, yeah, sorry. Yeah, so from the moment you get up from the bed and until you go to the bed in the night, you will be interacting with several goods, things in your life, like books, toothpaste, toothbrush, and you may be commuting or biking. Everything, if you look at it, the science is there behind those things, right? So without science, you cannot start your life, and without science, you cannot sleep your life. So would you like to be part of the scientific community? I'm sure you would like to become the part of scientific community, right? And in fact, you should feel proud of becoming part of scientific community. So hope you all would work towards becoming a scientist. So what you want to do in your life as a scientist, let's see. So what I have done it. Okay, so what I wanted to do and what I've done, and I work mostly as Dr. Pawan Madam told you that I worked on bacteria, and I worked on extremophiles. Okay, extremophiles means the bacteria which are living in extreme environments. What is it? I'm not able to see my titles. It's going to be trouble. It's not that big, so let's see, then it will
Sometimes
be.
it just goes away after it.
Okay. Okay, so what is this? Can you see?
Slide. Sir?
Can you see what is there on the slide? I'm asking the
We are
children.
able to see, but we are missing.
I'm asking children.
the-
Children, can you see what is there in the slide?
Yes, sir. Yes,
What is it?
Sir.
Okay, what do you know about this? It's a globe, right?
Yes, sir.
And I'm sure you know what is the globe made up of. Like it contains 71% of water and 29% of land, correct?
Yes, sir.
And if you look at it, the water component, out of 71%, only 3% is potable or usable because it's fresh water. And even out of the 3%, most of it is in the ice form or in the groundwater, right? So very less water is available. And also land, if you look at it, 50% of the land is not accessible to humans because they could be mountains or uninhabitable places, or whatever it is. Okay. And in such places, there are certain living things which are present. Okay. They are the microbes. Do you know how many microbes are there on this planet?
No, sir.
The entire globe is made up of microbes. You go to any corner, any corner of the globe you go, you will see microbes. So just to give you a number of how many microbes are there on this planet, can you see that number? See the number? It is 5 into 10 power 30. Mean after 5, if you put 30 zeros, that is the approximate number of microbes which are present on this planet. Now you compare that with our population. We are only 1200 crores on this planet, right? Imagine how many microbes are there and why so many microbes. What are they doing to the nature or to the globe? That is very interesting. And if you look at that composition, how they are distributed, see, in the open ocean, you see the number. Just one. Can you give me this? Is it okay if I show like this? Do they see? Or should I do I have to use that laser? Okay.
Oh, no, they'll not be able to see this. To read it.
Children, can you see the mouse?
Yeah, sir.
Okay, good. Yeah. See, if you look at the distribution of this 5 to 10 power 30 microbes on this planet, in the open ocean, there are 1.2 into 10 power 29 microbes. And even in the open ocean, if you look at it, the upper water, which is about 200 meters, and water below 200 meters, it is like this. There are 3.6 into 10 power 28 and 6.5 into 10 power 28 microbes in the ocean water. And besides the water, there is also sediment, right? Below the water. So the sediment also contains approximately 1.7 into 10 power 28 microbes. And below the ocean sediment also, there are bacteria up to certain, let's say, it depends up to 10 to 15 meters, and they contain 3.5 into 10 power 30 microbes. And that is about the water. And if you compare the microbes in soil, you can see that there are 2.6 into 10 power 29 microbes in the soil. And in the extracellular, if you take that, it is approximately 2.25 to 2.5 into 10 power 30. And if you take 1 gram of soil anywhere, and the approximate number of genomes found were about 4,000. Meaning, if you take just 1 gram of soil in your hand, that contains 4,000 different organisms. Imagine if you are making a house or dam, how much of disturbance you are causing to the nature. Because every genome, every cell, or every life has a purpose. The nature, in the evolution, the process of evolution, it has selected bacteria or any life form because of its function or its purpose. So when we do this, we are disturbing the nature, and that might lead to any disaster, and the consequences we cannot really estimate. For example, now we are doing too much of mining, taking iron from this end to other part of the globe, like the iron ore is being exported from Andhra Pradesh to Japan. What that causes to the nature subsequently in future, we have no idea. And this is what you people have to do it, because future is in your hand. Okay. And how much we know about this? If you look at the microbes, there are approximately, if you look at the bacteria, you can see that there are approximately 0.0 to 10 power 6 number of species. Okay. Out of so many species, as of today, if you look at it, there are close to only 30,000 bacteria or new species have been identified. And compare this number, 30,000 is nothing but 3 into 10 power 4, right? And we need to identify so many novel bacteria. And we don't know what is the function of those novel bacteria and how best we can make use of them. And if you look at the mammals here down there are close to 4,170 species, right? We know all of them. And if you look at the birds, we know all of them. The reason we know about them because they are morphologically very distinct. We can easily identify them, and we can also see them, right? Whereas bacteria, viruses, and archaea, can you see them? No, right? So there are special methods to. Yeah.
Sir, we are seeing only first slide, sir. I think you opened the file twice, sir. I think the file you opened, just close it. I think another file maybe you open.
Okay, that.
You still see the first slide only?
Yes, still seeing first slide only.
How do you see the biodiversity slide?
Yes, now we are able to see.
And the previous one?
That too in full screen. Yeah, previous slides we haven't seen, sir. Only first slide we see.
Oh, sorry.
వద్దు అన్నప్పుడు
Again we
నువ్వు
are seeing first slide only.
మంచిగా చెప్తున్నా వద్దు ఏ వద్దు పెట్టా
Can you see now?
Yes, sir. First slide.
Better in pictures.
Only first slide.
But now can you see this slide?
First slide only, sir. Previous slide.
That we are seeing, sir.
First slide, sir.
Do you not see the biodiversity slide now?
No, ma'am. Highlight.
Now can you see this slide?
No.
This slide.
No, sir. First slide only, sir.
We are seeing only first
Only
slide.
seeing first slide.
Yes, sir. Yes.
Now,
The same thing repeated, madam.
still see the first slide?
Yeah.
First slide. Previously
We
you
don't
did
know.
it, ma'am.
We don't know what to do here because here the slides are changing.
No, not at changing, madam.
From at our end, it is changing. So, shall we send you the slides?
Yeah, Somdata, please send the slides to me. To
Okay.
WhatsApp. I can share it with the host and she can share the screen.
Okay.
Yeah, Pawnee ma'am send.
Is it email on?
Yeah. Good. Yeah. No. This one. It's open it. Yeah.
After email is open.
Yeah.
It is account. Is it?
Got. This one.
Somdata, did
Yeah.
you share the presentation with me?
This is on Dr. GSN Reddy's computer, so
You can email it to me.
now
Put
the
my email in the chat box.
that is also okay. Put it. Do you have Gmail?
Just email it to me, or else I'll write one more email ID. My colleague's email ID.
Please do that.
Yeah. Yeah, that's my colleague's email ID. osd.acad2@gmail.com.
Just a second. He is logging into his Gmail because the government ID is not letting that big a file to go in. This. आपके फोन पे कुछ आएगा.
Yeah, it's osdacad2@gmail.com.
You're still struggling to. Is it on the? Is this a pen?
No, this.
There is no dot. It's just
Can
osdacad2.
you put that PPT on this and give
It's
me?
already there. If it works from there, maybe can I use that laptop?
Maybe.
Yes.
Can you tell me the email ID that company has
Okay.
on the chat box?
Where it promise you want?
Whatever she's given.
This one.
Please go to the Zoom. Go to chat. Click on it. OST.
OSDA. OSDACAD2 at gmail.com.
What else? She's given. Okay. So, Pavani, I'm sending it to the OSD attached to Gmail ID.
Yes, Somdata, please.
Okay.
Yesho ma'am, if you're on the call, please download the presentation and could you please share?
We can just try here whether we can do it.
I just sent the email, Pavani.
Yes, Somdat, I've asked Yasholata ma'am to take care of it. She'll present. She'll start sharing the slide.
We can just try
I
to.
don't understand why there is a technical problem today. I'm so sorry, sir.
Actually, I am feeling sorry for this.
I just got the email, ma'am.
Ma'am, అదే చెప్తున్నా అదే చెప్తున్నాను.
Pavani, can you please make me the host? I've just logged in from my own account now. And also enable my screen sharing. Yeah, perfect. Okay, so they are sharing the screen and you can speak from here. Yeah, I'll take care of this.
Oh, this is a full scan, right?
Huh, you. Speak.
Okay.
You'll need to tell them when to. So now you cannot change these slides. They are showing the slides.
Okay.
So you have to tell them when they have to go to the next slide.
This like this. Okay.
So should they stay on this slide or the next slide?
Whom should I tell? Like
You can
okay.
speak. They'll do.
Can you go to the first slide, madam?
We'll start from the beginning again.
Yeah. Okay. Now this I've already talked. Go to the second slide.
Yes, sir. Yes, sir.
Children, you have seen the title of my talk, right?
Yes, sir. Yes, sir.
So I'll be talking on exploring extremophiles. Okay. Can you go to the next slide?
Okay.
Yes, sir.
Yeah. So what I was telling is that I showed you a globe, and then what is the globe made up of? I showed you that the globe contains 71% of water and then 29% of the land. And I also told you how water is distributed on this planet, and also, you know, how much land is being used by us and how many microbes are there on the planet Earth. As you can see again here, there are 5 into 10 power 30 microbes. Okay. And when you look at the distribution of these microbes on the planet, can you go to the next slide?
Yes, sir. Yes, sir.
Have you seen this slide?
Yes, sir. Yes, sir.
Okay, yeah. So I was telling you what is the distribution of microbes in upper water, below 200 water, in the sediment and below sediment, and also in the soil. And I also told you that if you take 1 gram of soil, it contains 400 different genomes, meaning 400 different organisms which are present. Okay. Can you go to the next slide?
Okay, sir.
Yeah.
Sure.
Yeah. So this slide shows you that how much of diversity is known till today. If you look at the mammals, higher animals like mammals, birds, fishes, more than 90% of their diversity is known. Okay. The reason it is known because we can, morphologically, we can easily identify them and we can distinguishedly identify and then record them. Whereas if you look at the microbes like bacteria, fungi, viruses, and archaea,
not much is known because they are microscopic. So when they are microscopic, you cannot see with your naked eye, right?
Yes, sir.
Yes, sir.
Yeah,
Yes,
so you
sir.
need microscope, and even then, can you identify with the shape? No.
So we
You have to
cannot see with
No,
our
sir.
naked eyes.
Yeah, you have to look into the subcellular molecules to
Microscope.
identify
Under
them.
microscope.
Because of that, though there are 3 into 10 power 6 now species which are predicted on this planet, as of now, we could identify only close to 30,000, which is only 2.8%. Hello? Okay, so only 2.8% of it is known. The rest has to be identified because microbes which are evolved on this purpose, they evolved because of a purpose. There is a purpose for it. It has some contribution to the globe or the planet Earth. If you want
Sir,
them,
what
you identify them. And today, even after hello, can you hear me?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay. Thank you. Okay, yes.
Sir, we can't able your voice.
So there is only 2.8% of the bacteria population which is known till today. So the rest has to be explored, and which is in your hand. I get some disturbance.
Once again, sir, most of them in unmute mode. Please, all of you students, mute your audio.
Yes. Please mute your audio and video also.
Okay, can you share the slides, please? With me? Hello? Yeah, okay. Next slide. Yeah, okay. So yeah, you can hold on this slide, madam. So there are several microbes which are very, very interesting because they have some unique function, and also some of the microbes, as I told you, they have some purpose, right? And some of these microbes, like our animals, they're also becoming extinct. They're disappearing from this planet Earth because of human intervention. For example, if you take some of the microbes like Penicilliopsis, Cucania, all these microbes have disappeared because their habitat also has disappeared from this because of our humans' excessive harvesting of the nature. We are cutting down the forest. We are exploring the oceans for oil, and also we are
increasing vehicle pollution.
Yeah, exactly. Vehicle pollution, everything is causing the extinction of some unique and interesting, not only animals, even the microbes. And there are so many novel microbes, and some of them could be very, very interesting. I'll show you in the next slide. And if you look at their use or their potential in the industry, you can see that all these microbes can be used for enzymes, fatty acids, antibiotics, in bioremediation, and also biodegradation. I'll be touching upon this in my later slides. Can you go to the next slide?
Yes, sir.
Okay, sir.
If you look at this bacterium called as Thermomargarita namibiensis, this is the largest bacterium ever known till today. And if you look at its size, it is almost 3.3 millimeters, meaning if you compare the scale which you have, you can see with your naked eye. You don't need a microscope to see this particular bacterium. You see how big bacterium it is. It is a gigantic bacterium, isn't it? And it was found in Namibia, in the coastal sediments of Namibia. If people like, if scientists were not interested in exploring the microbes, we would have missed such beautiful and unique microbes, isn't it? And the smallest bacterium, if you see, the smallest one is called as the nanobacterium. It was found in the kidney stones. Have you heard of kidney stones? You must have, right?
Yes, sir.
So this was found in the kidney stones. What is it doing there in the kidney? What is its function? As I told you, the nature has selected every organism for some kind of purpose. So what is the purpose of this nanobacterium? Is it making the crystals in the kidneys, or it is trying to dissolve those kidney stones? We have no idea. Unless you people, people like you come into science and then do research and then see what is its function, we wouldn't know that. Got it? So hope you'll come into science, right? And besides this, if you look at the bacterium called as Deinococcus radiodurans, it can tolerate a very high amount of radiation. And this is very, very beautiful bacterium, because when it is exposed to the radiation, it will break its DNA into pieces. Exactly. Any number of times you expose it to the radiation, it will break the DNA only at that particular spot. And once you withdraw the radiation, again it stitches. How does such a small bacterium do? I'm getting disturbed. Can you?
Please mute yourself. Sir, please mute yourself, sir. Okay, sir. Sir, your voice is not audible. Please unmute yourself.
Can you please go to the previous slide?
Yes, sir.
Yeah. Okay. That's right. Okay. So I was talking about this Radiococcus. Hello. Yeah. So this Deinococcus radiodurans bacterium. Can you hear me? Can you hear me?
Yes, sir. Yes, sir.
Is a bacterium which can take any amount of radiation.
Sir, we can hear you, but there is a noise.
No, sir.
Yes, teacher. Disturbance, sir.
There is a disturbance in somebody's house. Some children are disturbing the students.
All of you mute yourselves.
Can you ask all children to mute their laptops and phones? Yeah, they have done it. Okay.
So we muted everyone.
Yeah. Okay. Okay, madam. Yeah. So this Deinococcus radiodurans, it can tolerate a huge amount of radiation. When you expose this bacterium to the radiation, what it does is it breaks the DNA into pieces. And when you withdraw the radiation, again it stitches back to the whole chromosome. And how does such a small bacterium know where to cut it and how to stitch it? It's an intelligent bacterium, isn't it? And just for comparison, if you see, when we go to the X-ray, we are given close to 1 milligray of energy, radiation. And if you are by mistake given 5 gray of energy, you'll be killed. But this bacterium, imagine, it can tolerate 5,000 to 50,000 rats. Isn't it not a big radiation energy? So there are certain unique and interesting microbes on this planet, and only thing is they are waiting for us to reach them and explore. Can you go to the next slide, please?
Yes, sir.
Yes, please, sir. Yes, sir. Yes, sir.
I can't see the next slide. Can you go to the next slide, please?
Okay, sir. Okay, sir.
Previous, Yeah, previous
sir.
one. Yeah. So if you explore these microbes, these unique ones and novel microbes, they can be used in various fields such as medicine, agriculture, forestry, fisheries, and biotechnology. How they can be used, I'll come to that little later. Okay.
Okay,
Go to the next slide. And as you see, most of the planet Earth, okay, which is not accessible, and even in them, some parts are very, very, very extreme because of various conditions. Okay, so the bacteria which are living there, they're from village, I guess. So the bacteria which are living in these extreme environments are called as extremophiles. Extreme means acute, and philes means philic means love, right? They love such extreme environments and they live in there. For example, if you take psychrophiles, which love cold. Psychro means cold. Philic means again love. So they love cold environment. Contrary to that, there are some bacteria which can love very hot temperatures, and they're called as thermophiles, which can tolerate a temperature of approximately 100 degrees centigrade. Imagine if you put your hand in boiling water, what happens? You get burns, right? Bubbles, right? This bacteria can happily live in there. It can divide. It can all its enzyme. It can do its life there. And there are certain bacteria which can live even at pH below 3. They are called as acidophiles. Are you listening to me?
Yes, sir. Yes, sir.
Yeah. So bacteria which can live below 3 are called as acidophiles because they love acidic pH. And contrary, again, there are certain bacteria which can live in very high pH, and they are called as alkaliphiles. So to say there are so many bacteria which can tolerate or live in extreme environment, and just to take few examples, I told you about psychrophiles, thermophiles, but there's so many like, halophiles which love salt, piezophiles which love pressure. Under high pressure, they can live in, like, in the ocean beds, there'll be so much of water on top of it, and still they can tolerate that pressure. Okay. And there are certain bacteria which can tolerate carbon dioxide. Can you people tolerate carbon dioxide? No, right?
No.
These bacteria love carbon dioxide, and they're called as capnophiles. Okay. So you can see there are so many types of extremophiles. Okay. So where do they live? First of all, you should know where they live, right? So can you go to the next slide, please?
Okay, sir. Yes, sir.
Sir.
Can you go to the next slide?
Okay, sir.
Yeah.
Okay, sir.
Yeah. So these are the extreme cold places. For example, if you take Antarctica, Arctic, the temperature is, sometimes
Very
can go
cool.
to, very cool. It can go to -80. There's a place called as Lake Vostok. There is a lake in Antarctica, and people have gone below that lake. That lake has approximately 8 kilometers thick ice. Okay. And below the ice also, under 80 kilometers depth, there is a bacteria called as bacillus. So, imagine how bacillus can live in under such huge chunk of ice. Is it not interesting?
Yes, sir.
Right?
Yes,
Yeah. And like Antarctica and Arctic, there are also some cold places in our country, in India. Okay, if you go to Himalayas, you would see them. For example, there is a lake called as Rupkund Lake. Okay, the temperature again there is around -40. Sometimes it can go to -60. And there are several glaciers where I have been to, like Pindari Glacier, Hamta Glacier, Kafni Glacier. From all these glaciers, we got the samples. Okay, and then we isolated the bacteria. Besides that, there's a huge, huge cold environment. It is the deep sea. As you go deeper, the temperature decreases, and the average depth of a sea is something like 2 kilometers, but the deepest place is around 4 kilometers. And at that depth, the temperature is below 2 degrees centigrade. Is that not a cold temperature? It is, right? So even at that temperature, microbes do live. Okay. So these are all extremely cold loving, and they're called as psychrophiles. Can you go to the next slide, please?
Yes, sir.
Yeah, like extremely cold or loving bacteria, there are some bacteria which can love extremely hot environments. For example, if you take on your right, the water is coming out from the soil. It's called as the geyser. And there are certain hydrothermal vents where the hot water is always coming through those vents. Okay.
Hot spring.
Hot spring, exactly, good. Okay, so the temperature of those is, it can be anywhere between 45 to 135 degrees centigrade. And such places are also there in India. Have you heard of Himachal Pradesh?
Yes,
In Himachal Pradesh, there is a place called as Manikaran, and there is a hot spring, and the temperature of that is very high, where people can cook rice. Can you see the one person keeping the pots there? So he can cook rice in that hot water. He doesn't have to do anything. Just take the rice in the pot, put it there, and it will get cooked. Such hot springs are there, and even in those hot springs, you would see bacteria living. Okay. Can you go to the next slide, please?
Okay, sir. Okay, sir.
Can you go to the next slide?
Okay, sir.
And these are the extremely salty places. In India itself, there is a place called as Ramba Lake. And in that lake, the lake water contains approximately 4 grams per 100 ml. 100 ml. That is, it means 4% of salt is there in that lake. And people are harvesting the salt, and we are also eating that salt from there. And if you go to a place called as Gittel Pond, this is the highest salty, saline lake, which is present in Ethiopia, and the salt concentration is approximately 4.5%. There are close to 1,000 salt lakes on this planet Earth. Imagine, so many halophiles are there, and these bacteria, they love salt. Okay. Can you go to the next slide?
Yes, sir. Okay, sir.
Yeah, and these are pH loving bacteria. If you see the 3 slides above, okay, and the first one is Lonar Lake, which is present in Maharashtra in India, and that lake pH is around 8 to 11, and even at that pH, bacteria can live. We have isolated bacteria from this Lonar Lake, okay, and most of them, they can easily tolerate pH 11, and these bacteria are called as alkaliphiles. Alkali means high pH, again philic means love, right? Alkaliphiles. And in contrary, again, there are acidophiles which love acid environment. There are certain volcanic acid drains. Yeah. In all these places, the water contains, very low pH, sometimes below 5, and bacteria can live in there. Do you people know that even in our stomach, the pH is very, very low? It is less than 2.5. And the bacteria which lives in there is called as Helicobacter pylori. Have you heard about this bacteria? You must have, right?
Yes, sir.
Yeah, so these are all acidophiles. Can you go to the next slide, please?
Okay, sir.
Yeah. So besides on the planet, there are also bacteria present in the outer atmosphere. Okay. There is a theory. Can you off that? Yeah. Okay, so the microbes are also found in outer atmosphere, and there is a theory called as panspermia, and according to this, a group of people believe that the life has originated somewhere extraterrestrial, and it was brought to the earth by dust particles. Yeah, so every day, sorry again. So every day, close to 200 tons of dust is being added to our planet Earth from other planets. So during this process, the dust may be bringing the life from outer space, from other planets, or meteorites or stars, and then depositing it on our Earth. And maybe that's how the life must have evolved. So it is not. It is not amazing that to know that the microbes are also present in outer space. So, but how do you explore them? I mean, if you want to explore them, you have to get the samples, right? So how do you bring the samples? You can bring the samples when one particular habitat is accessible to you, right? The places where you can go and bring it, you can bring it. But deep oceans, deep ocean water and outer space, can you get it? Difficult, right?
No, sir.
But there are ways and means to get those samples also. Can you go to the next slide, please?
Okay, sir.
Next slide, please.
Yes, sir.
Yeah, if you see here, soil samples can be easily collected with a scoop in a sterile bag, and
Yes, sir.
lake water and all, you can
Yes,
just
sir.
take it with a sterile spoon or scoop. And if the lake is not very deep, you can also get water samples using a bottle called as a camera. Okay, this is possible only when habitats are accessible to you, where you can reach, right? And if you
Yes.
want to get the little deeper samples, like core samples from the earth, there are corers which can go up to 5 meters, and then you can get the sample from there. And if you want the sample from your gut, from your stomach, how do you get it? There is a tool called as endoscopic tool. You can send this tubing inside your stomach and get the samples, and then you can study that. Okay, but these are all possible only when habitats are accessible to you, the places where you can reach. But how do you get it from places where you cannot reach? Can you go to the next slide?
Okay, sir.
Can you go to the next slide, please?
Yes, sir.
Can you go to the next slide?
Okay, sir.
Yeah,
Okay,
thank you.
sir.
Yeah. So
Yes, sir. Yes, sir.
Okay, sir.
if you see the picture on your left, okay, there is a bunch of tubes. They're called as Niskin sampler tubes. These tubes are sent into the ocean, okay, and then you can collect it at wherever you want it. Like any depth, you can take it down, and then you can close it. The water will be collected by that, and bring the water up, filter it, and then look for the microbes. Okay, so this is for the deep water sampling. These bottles can be used, and there is a corer. Can you go to the next slide?
Yes, sir.
This kind of corers can be sent to the bottom of the ocean, and then again you can get the soil samples. Okay, the sediment samples.
Okay, sir.
Can you go to the next slide?
Yes, sir.
Yeah. So if you want to collect the samples from the outer atmosphere, how do you do it? This is called as a payload. It contains certain tubes. They're made up of stainless steel. Okay, they are UV irradiated. They're absolutely made sterile. Okay, and they will be sent up using a balloon. Can you go to the next slide?
Okay, sir.
Yeah, you can see that the balloon, the one which you see on your left, it's a huge balloon. Okay, the balloon is approximately 40 feet or so. Okay, and it can be sent to different heights. It is filled with hydrogen gas, and whatever heights you want, you can open those tubes and collect the air samples, and then bring it back, and then you can analyze. Okay, so these are the methods which can be used for collecting the samples. So after collecting the samples, what do? You have to analyze, right? So how do you do that? You have to look for the bacteria, which are especially very, very extremophilic, right? So how do you do that? Can you go to the next slide, please? There are two ways to analyze these samples. Okay. The first one is called as a non-conventional method, where most of the bacteria cannot be cultured in the laboratory. The reasons are many, because we don't know anything of those microbes, what they eat, what kind of food we have to give it to them. We don't know, right? We don't know what are the conditions they require.
Yes, sir.
Such bacteria, whatever you do, you cannot immediately culture them. So such bacteria, we cannot really ignore it, but there are other methods which I'll show you in my next slide. Okay, that method is called as a non-conventional method or non-cultivable method. And the other method is called as the conventional, where you can culture the bacterium on the synthetic medium. Okay, because many times we may not be lucky, because if you take any sample, only 1% of that particular sample contains the cultivable bacteria. Rest 99%, they cannot be cultured in the lab, and you have to go to the non-conventional methods. Can you go to the next slide?
Okay, sir.
Yeah. So as I told you that 99% of the bacteria cannot be cultured, but still we can study what they are. We can identify them on the basis of a molecule called as a 16S rRNA gene. So what you need to do, you take that particular sample, isolate the DNA, you can see there. Okay, from the DNA, using 16S rRNA gene primers, you can make the 16S rDNA. Okay, rDNA, then sequence it and then you can identify. Once you know what it is based on 16S RD in a sequence, you go back to the literature and see what it eats, what kind of medium it requires, what kind of conditions it requires. Okay, you would know from the literature, and based on that information, again you can go to the conventional method and then culture them. Okay, can you go to the next slide?
Yes, sir.
And
Yes, sir.
see, this is how the 1% of bacteria which can be brought into culture can be grown. Okay, it's very simple. You take any sample, suspend it in water, plate it on a suitable medium which requires that particular bacteria, incubate it, and you'll get some colonies like this on a Petri plate. So we use a plate called as a Petri plate. It can be made up of a glass or a plastic. You put the medium in that. Medium is nothing but the bacterial food, and bacteria will grow. Now you can see on this slide, there are so many colored, dots, all those bacteria are there. And now you have to purify them, means you have to separate that particular bacteria individually. And if you see the next slide, that's how they got separated. Once you know that they're separated and they're pure, then you can use them for biotechnological applications. Several things can be done. Okay. And what kind of media you need to require? Again, it depends on the type of bacteria which you are interested in. Can you go to the next slide?
Okay.
Okay, sir.
Yeah, if you see this slide, the bacteria requires different conditions. Okay, the condition depends on chemical requirement of chemicals or the light. If they require light, they're called as the phototrophs. Like plants, they're autotroph, right? They depend on the light, right?
Yes,
like
sir.
that, there are also bacteria which depend on light. Okay, they're called as
Some of the
phototrophs.
bacteria, not all bacteria.
Yeah, cyanobacteria, that kind of bacteria, they need light. Okay,
Living
if you are
bacteria.
interested in that, you are exposed to light. And there are certain bacteria which require chemicals, not the light. They are called as the chemotrophs. Again, these chemotrophs and phototrophs, they can be classified based on the carbon source, whether they need carbon source or they don't need carbon source. The phototroph which require carbon source are called as photoheterotrophs, and which don't require the carbon are called as photoautotrophs. They take light and make their food, like cyanobacteria. There are certain bacteria, they need light, but again, they also need the carbon for their food. Okay. And similarly, even the chemotrophs, they also require carbon, then they are called as chemoheterotrophs. And there are certain bacteria which can make their own food, again, using some chemicals, they are called as chemoheterotrophs. Okay. These are the basic conditions which you have to keep in mind when you are going for isolating, type of bacteria you are interested in. Again, as I told you that depending upon the extremophilic bacteria which you are interested in, you have to change the conditions. Suppose if you are interested in psychrophiles or thermophiles, you have to consider the temperature as a parameter. And as for acidophiles and alkaliphiles, pH, high level salt, and for anaerobes and anaerobes, if you are interested in, you don't have to use oxygen. Okay, these are the conditions you have to keep in mind while you are isolating the bacteria. Can you go to the next slide? Okay. So this is the outcome of your isolation of extremophilic bacteria. This is the amazing bacterium which has revolutionized the entire world in the field of molecular biology. Without this particular bacterium, the microbiology or the molecular biology wouldn't have been there. Have you heard of RT-PCR tests? You must have, right? They do RT-PCR test for diagnosing COVID.
Yes.
So what is RT-PCR? Have you ever thought about it? No, right?
Reverse transcription polymerase.
Good job. Yeah. So in that PCR, they use a particular type of enzyme called as Taq polymerase. It's a DNA polymerase isolated from a thermophilic bacteria. Who isolated that particular bacterium? Two scientists like you, Dr. Thomas de Brock and Fries.
Thomas de
Brock and Fries, they went to Yellowstone National Park in America, and they saw these hot springs, and they were also amazing to see this, and they were curious to see what is there in that hot spring. They got the samples from there and they tried to cultivate by mimicking those conditions like temperature, pH, everything in the lab. And to their luck, they got some bacteria, out of which they identified one bacterium as Thermus aquaticus. And again, they were curious to know how it is surviving there. Can we survive in hot springs? No, right?
No.
We get killed.
Yes, sir.
But if it has to survive, what is central dogma? There are three processes, right? Replication, transcription, and translation. All these processes have to be taken by the bacterium to live in there, right? So they looked into this, and while doing that, they identified one particular enzyme called as DNA polymerase. Where is this enzyme useful? It is useful in replication of DNA. Okay, so that particular enzyme, when they characterized, they found that it can tolerate a temperature of 100 degrees centigrade. Nothing happens to the enzyme when you heat it to 100 degrees centigrade. Okay, and it can work optimally, means the highest function they have observed for this enzyme at 72 degrees centigrade. Okay, because of these characteristics, Dr. Mullis has developed a technique called as PCR, where he has used this enzyme and then developed this technique, and now this technique is being used in every molecular biology lab, every diagnostic center. Okay, and you also become very rich like Noble. You don't want to make dynamites to smash people, but you can make such enzymes, identify such enzymes, and become very, very rich. Okay, got it? Is it not interesting to identify such microbes, unique ones?
Yes, sir.
It is, right? Yeah. Can you go to the next slide, please? Yeah, this is the bacteria which I have identified. It's very interesting. If you give good food, rich food, it will grow in pink color. Why that pink color? Because it makes a pigment called as carotenoid. Okay, you can see the beautiful color in that. But if you starve it, okay, if you don't give that food, it is not very unhappy and it will still try to grow. How? It will make a pigment called as melanin, which is black in color. These are the spectrum. When you go big, you would understand the spectrum. But now for time being, keep in mind that under low nutrient conditions, it will grow as black. And when it is rich in nutrients, it will go as pink. And this melanin is very, very, very useful because it will work as a sunscreen. You can nicely put it on your skin to avoid exposure to UV radiation. You can tolerate the UV radiation. And also this particular pigment is antifungal. It will protect not only from the light, but it also protects you from fungal infections. So if you make such pigments, you can sell it and again become very rich. Can you go to the next slide?
Yes, sir.
And these particular proteins called as ice nucleating proteins, they were found in Antarctica, secreted, are made by bacteria. Okay, and what do these ice nucleating proteins do? Suppose when you keep a glass of water without disturbing the fridge, it doesn't freeze. Have you ever observed that? When you just give a slight jerk, like that, it will form the ice. Okay, so why? For formation of ice, you need at least one crystal so that all water molecule can crystallize on that particular crystal. So these proteins, they do that job in Antarctica and help the Antarctic environment by making growing the ice on it. Okay, so such ice nucleating proteins can be used in making ice, and this particular protein is being used in skiing, where go to the Himalayas or places where there is too much of ice for skiing, right? So if you want to make such artificial ice skiing parks, you can use this protein. Just spray this protein and try to cool it, and the ice will just cool immediately. In contrast to that, there are other proteins called as antifreeze proteins. Can you go to the next slide?
Yes,
and
sir.
this particular antifreeze proteins were found in Antarctic fish. How Antarctic fish live in there, even though the temperature is very cold? Because of these proteins. You know that there are cold-blooded and warm-blooded animals, right? Yes. We are warm-blooded animals. We always, our body maintains 37 degrees centigrade.
37 degrees.
Exactly. The cold-blooded organisms, they maintain the temperature of the environment. So when the Antarctica temperature goes to -40, like water, the blood also should freeze, right? Right? But this particular proteins are there, and which will prevent the formation of ice in them, and they make fish and other organisms live in there. And this protein can be also used in where, in cold countries, when you want to supply the water, in cold countries, the water will freeze in the pipes, right? So using these proteins, you can avoid freezing of water in taps in the communities. Okay. Is it not very interesting to look for such proteins in bacteria also? It is,
Yes.
right? So now it is in your hand to come into
Yes, sir.
science and explore such unique bacteria. Can you go to the next slide?
Yes, sir.
You can see there are a couple of proteins which I've listed here and their applications. All these proteins were isolated from Antarctic bacteria. They're very, very interesting. They have a lot of applications. You see lipase, if you see lipase, it has application in food, detergent, cosmetics, several applications. So all these enzymes, if you make it in bulk, you'll become very rich. You can establish industries, right? So the science is only waiting for you, for people like you. Can you go to the next slide? Yeah, so these are the challenges. These are the challenges which you people have to take it up. Okay. First one, if you see the human health. Now you know COVID, right? Why we are affected by COVID virus? Is there any way to prevent it? Yes, there are ways. It is known that when viral sample is injected into mice with a probiotic bacterium like lactobacillus or bifidobacterium, the viral load decreases. So if you can identify a good probacterium and then give it or put it in your stomach or in the gut, okay, so that what happens, the result is you'll not get infected from this kind of viruses, right? So now your job is to identify a good probacterium, okay, which is suitable or which can prevent the other bacterial or viral infections. That is in your hands. And again, treatment. Have you heard of septimiasis?
Yes, sir.
Right? What happens? Your blood gets poisoned, right? And
Yes,
within
sir.
2 hours of the serious infection, if the patient is not treated, then he will die. So, but how do you identify? There is no diagnostic method as of today where you can identify a causative bacterium of a particular disease in less than 2 hours. The regular process is send it to the diagnostic center, somewhere, they culture it, they do the antibiotic screening, and again, takes 2-3 days to identify that particular bacterium, and then the antibiotic profile of that particular bacterium. And the time taken is used, and meanwhile, the patient will die, right? So now you have to develop the diagnostic techniques where you should be able to identify any disease-causing bacterium or virus in less than 2 hours. Okay, and then probiotics, as I told you that again, there are several probiotics which can help in prevention of viral infections, right? As of now, we know only lactobacillus and bifidobacterium. Is there any other bacterium in the gut which we have not studied yet, because human microbiome is just coming into picture only now, their impact and all that, which I'll be talking it in later slides. So we don't know much about it. So you just look at the very good probiotic bacteria or bacterial consortium, which is useful for us. And again, if there are bacteria which can make antibiotics within your stomach, they can kill the disease-causing or pathogens, right? Pathogenic bacteria, right? Is it not possible? It is possible, but only the thing is we have to work on it, okay? And then take up the challenge, and then we have to get such bacteria. And next one is the severe problem. It's called as an antibacterial resistance problem. What is happening? You must have seen your parents, or you also must have got the antibiotics from any medical shop without any prescription.
Yes, sir.
Right?
Yes, sir.
So this is called as the indiscriminate use of antibiotics. And many a times, because of financial problem, we don't use the full course of antibiotics. So we stop in between. Because of that, what is happening is that the bacteria is getting the resistance to such antibiotics. And whatever antibiotics are available in the market as of today, all these bacteria, once they become resistant to that, we cannot do anything. So now what are we going to do? You have to look for some alternate antibiotics, okay? Right? So now these antibiotics can be peptide based, which are secreted by bacteria. So you have to look for such bacteria which make this peptide antibacteria, because the bacteria cannot mimic itself to become resistant to such peptide antibiotics. So you have to look for such peptide antibiotic producing bacteria and then use them. Once you identify such bacterium and identify such antibiotics, again, you can become very rich. You don't have to be a doctor or a lawyer or a politician. And again, in agriculture, now you know that most of our farmers, they are using chemical fertilizers, right? You take urea, you take phosphate, you take anything. Can't we replace them? There are bacteria which can do that. There are certain bacteria which make the enzyme called as phosphatase. That phosphatase enzyme solubilizes the phosphate from the rock, from the soil. You don't have to give anything. Just give that bacterium there, it does its jobs and increase the crop production. There'll be more yield. Similarly, you must have heard about nitrogen fixing bacteria, azotobacterium, rhizobium, right? So they fix the nitrogen. There are nitrococcus, those bacteria fixes the ammonia. There are several good bacteria. Only thing is you have to identify the right bacterial consortium and then use it as a biofertilizer to increase the yield. Okay, and you'll get organic food. Similarly, biopesticides. Have you heard of a protein called as a crystal protein from Bacillus? Bacillus thuringiensis is a bacteria which makes a protein, a crystal protein, which will kill the mosquito larvae. Okay, there are several such bacteria. No? There is another bacteria which Australians have identified. It will prevent the fertilization in mosquitoes. If you spray such bacteria, then there are no mosquitoes, right? So there are beautiful bacteria on this planet Earth. Only thing is we shouldn't be greedy, just explore them and then use for our good. And forestry, you must have seen so much of wood which is being cut. Who's going to degrade that? There are the hardest material, lignin, cellulose, all this material. Who's doing that? You have to put that into Newton cycling, right? So only bacteria can do that. Bacteria, some of the bacteria have the enzymes called as ligninase, cellulase, chitinase. All these enzymes can degrade such hard materials and bring that into the Newton cycling and then fertilize the forest lands. Okay, so you can think of that. And dairy, farmers, they inject one hormone called as oxytocin to increase the milk production. Have you heard of that?
Yes, sir.
Yes.
Yeah, so that is an invasive method, right? What if oxytocin, after all, it's a hormone, right? If you clone that hormone in a bacterium which is resident of a buffalo or a cow, that can just make the hormone, and that hormone go into the system and then gives the milk, right? Why don't you think of that? Why you have to use such chemical molecules and then, use the invasive methods to make the cow or buffalo unhappy? So you can increase the milk production by looking at such bacteria, probiotic bacteria, which is there in milk, in cow's guts, right? Or in buffalo guts. You can do that. And poultry, have you heard of a bacterium called as a spirulina?
Yes, sir.
Yeah, it's a
Spirulina.
cyanobacterium. You don't have to do anything. You just take a tank, fill it with water, keep it on your terrace, put this bacterium, it will grow by itself. Every now and then scoop it out, and you can make a curry or something out of it, or you can give it to your cattle, or you can give it to your chicks, or in the poultry also can be used. This particular bacterium has more than 85% of protein. And you're supposed to eat protein-rich food, not the carbohydrate-rich food, right? So one way of getting the protein-rich food is that you can simply get that spirulina bacterium. Not only spirulina, there are some certain other cyanobacteria, cyanobacteria called as phygomyces. Even that bacteria also has very high amount of protein, and such bacteria can be used in the dairy or in the poultry feed. And coming to the organ synthesis, have you heard of an antibiotic called as rifampicin? When a person is infected with tuberculosis, this particular antibiotic is suggested by the doctor. And this particular antibiotic, I don't know whether you know the racemers. Have you heard of the racemers, all these isomers? Okay. So one form of this antibiotic is harmful. It causes the blindness. But other form is good and it will kill the tuberculosis causing bacteria. And if you separate the bad compound from this particular rifampicin, nobody can do it, not even chemically. Only bacteria can do that. There are certain bacteria which can eat up such isomers. And some of the enzymes also makes enzymes called as isomerase. They change the conformation. So there are several enzymes which can be used in organ synthesis. And biotechnology, vaccines, enzymes, antibiotics, proteins, all these can be made use of for our betterment. Okay, you can overexpress it, and then you can use them. And the last part of it is pollution control. how do you control the pollution? There's so much of carbon dioxide being emitted by vehicles, all that, right? Is there any way to do that? Bacteria can do that. Only thing is, you have to have the water bodies. You should not occupy. You should not spoil the water bodies. What happens if there are water bodies? Water bodies have all the nutrients required for the cyanobacterium to grow. When cyanobacterium grows, it sequesters the carbon dioxide from the environment and fixes in the lake, right? And ultimately, when the cyanobacteria dies, it goes into the bed. So bacteria can also be useful in converting this kind of emitted fuels into, into solid carbon, and then fix it in the ocean bed or in the lake bed. And another thing is, you must have heard in the recent news that Delhi is being polluted because of the paddy fields which are burned in Punjab or Haryana, right? Can't we make use of that, grass? That's, after all, it contains cellulose and some amount of starch, and it is already known that cellulose and starch can be converted into alcohol. If you develop a technique, if you can identify a bacterium, an optimum or efficient bacterium which can convert starch or cellulose into alcohol, our country's most of the energy problem is solved, right? So the energy problem or the economy of our country is in your hands. Identify such beautiful bacteria which can convert these metals into alcohol, and then you can commercialize that. Okay. Can you go to the next slide? So this is all about my talk, but I thought I also would touch something about our human microbiome. Okay. So human microbiome is, the latest work which has been done. It is evolved only after, evolution of the next generation sequences, where you can do the, sequencing efficiently. So after that, this has come out. If you see the number of cells in our body, there are only 37.2 trillions. Compared to that, our body carries close to 100 trillions, means 3 times our own cells. Okay. So what are those microbes doing on or in our body? They have some purpose, I told you, right? So it is in your hands to see what kind of purpose they have, how they are associated, what good they are doing it, or what bad they are doing it. Okay. Suppose if you take an average human microbiome, every human carries approximately 4 kgs of bacteria with him. So if your weight is around 50 kgs, assume that 4 kgs means your weight is only 46 and 4 kgs belongs to the microbes. So what are they doing? Okay. We have only 23,000 of genes, but if you look at the microbial genes, imagine how many genes they have. They have 33 lakh genes. Okay. And what is their contribution to our body? I mean, of course, they're also helping their their growth and other things, but they're also doing some kind of help to us, right? What are the genes which are helpful to us? You identify them, okay, and make use of them. For example, you go to the next slide. Can you go to the next slide?
Yes.
Yeah. If you see here, there are 3 types of diseases I've listed which are linked to the microbiome. Suppose if you see the pie diagram on your right, okay, in the healthy control, you see that pink and yellow one, okay, and it is changing in the patients where they have the inflammatory bowel disease, right? So what is happening? The major portion of this bacteria is formicutes, and I can see that below, and bacteroidetes, right? Their proportion is changing it into this shape, and they're causing the inflammatory bowel disease. Suppose if you can use the probiotics, or if you change the microbiome to the left, then you gain back your health, right? Similarly, even in type 2 diabetes, you see that the change in, the proportion in formicutes to bacteroidetes, it is causing the diabetes. Now, the bacteria, you develop a consortium and then change the composition, then you'll become non-diabetic. Meaning, how much of money you can save by doing that? 30% of our country population is diabetic now. So if you can develop right consortium or right probiotics for diabetes, that's it. You will become very rich, and that's a great achievement. Okay. So these are some of the things which can be done, and it is in your hand. You are very young guys. So take up science as a career, work on extremophiles, and then come out with such beautiful enzymes, whatnot, everything which comes from the bacteria. Can you go to the next slide? Yeah. I'm thankful to the director, Somdatta and Pawni for giving me this opportunity. And I would have really loved to have the physical interaction, but nevertheless, I enjoyed this talk, and I hope I've given you some kind of information which is useful for you, not only to teach to the students, but also to the teachers. Thank you. Any questions?
Thank you, sir. Thank you very much.
Sir, I have a question. Can you answer, please?
Sure. I can't hear you.
Sir, I have a question. Can you
Yeah,
answer that?
Yeah. Tell me. Ask me if I can.
Sir, how a virus can stay dead outside, and how it can stay alive when it enter the body?
You cannot really say that the virus is dead. It is active or not active. As long as it has its genetic material covered with protein, it requires only a host to multiply. Okay, so it is not dead, but as soon as it sees its host, it gets activated and then it will become live. So it's not that it's not really dead outside, but it is not active. Is that okay?
Thank you, sir.
Sir, I have a doubt, sir.
Tell me.
Yes, go ahead.
How did microbes get there in the first place, sir?
Say again.
How did microbes get there in the first place, sir?
Okay, see, microbes are there everywhere on this planet, not only on the soil, inside the soil, everywhere. So the nature selects. It is like, when you go for 10 interviews, you'll get selected only in one company, right? Where they like you, where they like your profile, right? Similarly, even bacteria also was selected long back like that. Once upon a time, before Gondwana period, I'm sure you know that the earth was a single pot, right?
Yes, sir.
After that, the earth got separated.
Okay.
And now you got 7 continents like Antarctica, Arctica, all these that. Okay. So once upon a time when earth was united, all bacteria were distributed. But as they moved, selectively only some were selected because Antarctica moved to south and it has become very cold, and whatever can survive at that temperature, they survive. It means what the nature has selected. Unfortunately, if such bacteria, such cold bacteria was selected by hot springs, you do not have survived, right?
Yes,
So
sir.
it's not that the bacteria has gone there, but the nature has selected by itself because they are present everywhere.
Okay.
Sir,
Yeah.
is there any special process which is involved in growth of microbes, sir?
Yeah, every bacterium, depending upon its conditions, okay, you need special growth requirements like temperature or pH or whatever, even salts, all that. You cannot grow all the bacteria on the same food.
Okay. Sir, the bacteria, how they are entering in our body, sir?
It's very easy, through food, through air, through water. Right?
Sir?
Yeah.
Yeah.
Sir, what are the characteristics of microbes, sir?
The very basic characteristics all microbes have: the membrane, cell wall, right? They have certain enzymes in them and genetic material.
Sorry. Is the spirulina, yes, sir. Spirulina is an bacteria.
Yeah.
Sir.
Is it bacteria or algae?
It's algae or cyanobacteria, blue-green bacteria. It's autotrophic bacteria, basically.
Yes, sir. It produces single cell protein.
Yeah, exactly. Yeah.
Sir, it is a microorganism.
Yeah. Whatever you see with a microscope, you can call it as a microorganism.
Yes, sir.
Sir, I'm Dhanush from Balnar School.
Okay.
Is bacteria cannot survive in high temperature?
There are certain thermophilic bacteria, they can survive. I showed you, right, that Thermus aquaticus.
Which food take bacteria grows, sir?
Pardon?
Which food takes bacteria grows?
Say again.
Student, sir is a
bacteria
Telugu
growth.
speaking person, so if you're not able to articulate the
You
question
can
in
also
English,
ask
you
me in
can ask in
Telugu.
Telugu.
Telugulo కూడా అడగొచ్చు మీరు.
బ్యాక్టీరియా
మెల్స్ కాదు.
పెరగడానికి ఏ యొక్క ఆహారం తీసుకోవాలి?
బ్యాక్టీరియాకు ఇంతకుముందు ఒక స్లైడ్ లో చూపించాను. వాటికి ఆక్సిజన్ రిక్వైర్మెంట్, కార్బన్ రిక్వైర్మెంట్, సన్లైట్ రిక్వైర్మెంట్ బట్టి మనం డిసైడ్ చేయాలి అన్నమాట. అంటే ఇప్పుడు నీకు ఇంట్రెస్ట్ ఉందనుకో ఫోటో ఆటోట్రాఫిక్ బ్యాక్టీరియా మీద, అప్పుడు నువ్వు ఓన్లీ సన్లైట్ ఇచ్చేసి కార్బన్ ఇవ్వాల్సిన అవసరం లేదు. ఓన్లీ బ్యాక్టీరియా గ్రో కావాలంటే మనకి కార్బన్ కావాలి, నైట్రోజన్ సోర్స్ కావాలి, ఫాస్ఫేట్ సోర్స్ కావాలి, రైట్? సో ఇఫ్ యూ ప్రొవైడ్ దీస్ and then sunlight, bacteria can grow if it is photo autotrophic. Atlane manaki, yeah.
Why can't some microorganisms can't be in high temperatures, sir?
Bacteria high temperature, okay, you know Telugu, right?
Yes, sir.
Yeah, if the bacteria has to grow in high temperatures, first of all, who sees the temperature? The cell membrane, right? So the cell membrane has to be rigid. How it can make it rigid? Because the membrane has some lipid molecules and also fatty acids. If you compare the bacteria which is growing at high temperature with the bacteria which is growing at low temperature, the bacteria which are growing at high temperature have long fatty acid chains, fully saturated.
Doubt.
Thank you, sir.
Yeah, because of these molecules, they can tolerate high temperatures.
In water, the microorganisms are present, no, sir?
In water, yes.
Then in boiling water, the microorganisms, while boiling, the microorganisms will dead.
Will die, correct. Because these microorganisms are not from the hot springs, right? They are not adapted to the hot temperatures. If you get the bacterium from a hot spring and then boil it in your house, it doesn't die because it is adapted.
Thank you, sir.
Sir,
Yeah.
Sir, what is the food for microorganisms?
Say again.
Sir, I have a doubt, sir.
Let me answer that previous question.
Sir, I have a question, sir.
Sir, what is the food for microorganisms?
Yeah, this question was asked by somebody else. Again, I'm telling you, it depends on the bacterium. If a bacterium is a photoheterotroph, you have to provide carbon, light, nitrogen source, ammonia source, and phosphate. See, if you really think what are the atoms with which our bodies are made or our cells are made, just tell me.
Sir, how microorganisms will increase?
No, answer my question, please, one of you. What do our cells contain? Our cells are any living cells.
nucleus.
That's an organ, but at the atomic level, what are the atoms it is made up of?
Cells.
Sir,
Very simple. Only five, right? Oxygen, right?
Carbon,
nitrogen, hydrogen, sulfur. Water again contains hydrogen and oxygen, yes, right? These are the elements which are present. Okay, so we have to provide these elemental sources in the food form. Okay, so bacteria can either use carbon or make carbon. Like plants, some bacteria can make carbon, so you don't provide carbon there. Some bacteria cannot make, so for them you have to provide carbon source. And you are providing phosphate because your DNA requires, because DNA has phosphate molecules, right? And some bacteria, they can fix nitrogen from the atmosphere. Okay, even if you don't provide, it will take it from the atmosphere and uses it and then fixes it. So it depends on the bacterium. Okay, based on that you can use different nutrients. Yeah, you got it?
Yes, sir.
Yeah, next question.
Sir, how can the bacteria which is present in atmosphere can resist the UV radiation?
Because again, it's an adaptation, adaptive process, adaptation process. Like, suppose if I put you in the cold, what do you do initially? For sometimes you'll wear a sweater, right?
Sir.
With the time you'll get used to it and then you'll remove the sweater also. What is happening? Your cells are getting adapted, acclimatized to that weather. So even bacteria also do the same. Yeah.
Sir,
Yeah.
does everyone have the same microbes?
Everybody has, yes. On their body and inside their body. Without microbes, we cannot survive.
Sir, microbes are grown, sir.
Yeah, you can grow them in the lab. They do grow in their environment.
Sir, how do microbes affect an environment?
Say again.
What is a microbiome, sir?
A bunch of microbes is called as a microbiome.
Sir, give two examples of biodiversity, sir.
Yeah. Tell me, you can speak in Telugu also, no problem.
Give two examples in our daily life of biodiversity.
Biodiversity, examples. You see diverse people, diverse vehicles, diverse animals with your naked eye, right? You have to see how many plants. When you go to your school, on the way, you see how many different plants are there, then you would understand. Right? That is the diversity. And in fact, every day you should, as the way you are using the, scientific tools, knowingly or unknowingly, like, toothbrush, toothpaste, your clothes, even your clothes, have you ever thought of what is made up of? What are your clothes made up of?
Variations.
You tell me.
Differences.
Tell me mostly what type of clothes you wear?
Cotton.
Cotton, good. Where does the cotton come from?
Cotton.
Where does the cotton come from? Plants.
Plants.
Why don't you get that from the animals? Do you get cotton from animals?
No, sir.
What do you get from
No,
that?
sir, it is not possible. Woolen, sir.
What is the
We'll
difference
get
between, you also wear woolen clothes, right? So what is the difference between cotton and woolen? Wool?
wool come from
Cotton
animals.
is plant fiber and woolen is animal fiber.
Good. But what is the material made up of?
Sir, cotton is made up of cellulose and woolen is made up of proteins.
Good job. Good
Beta
job.
carotene.
Correct.
Yeah.
You see.
Keratin. Yes.
No, that is the pigment which is there, but mostly it is made up of protein. It's the proteinous material. Okay. So every day, if you see something, you have to think at the molecular level. How cotton is made into a fabric? Can you simply make it? Is it possible with your hand? There was a lot of science went behind that, right?
Yes,
You
sir.
have to harvest the cotton, you have to remove the cotton seeds,
Yes,
you make it so many threads, all that. It's not possible without a scientific invention, right? Everywhere, whatever you see, drink, eat, everything, there is a science behind it. If you really keep thinking it, there's a lot of it. Without science, we cannot survive. We have no life. So that is the importance of science. So unless people like you come into science, explore so many things, we cannot. I told you there are so many challenges, right? If you want to get good organic food, you have to develop organic fertilizers, pesticides. It is in your hand. They are all there in the nature. Only thing is you have to get it out.
Sir,
Yeah.
high yielding varieties are seeds or chemicals, sir?
Both. If you put some chemical, it can do it. For example, plant growth hormones are there, right? Auxin, gibberellins, all these. These are all chemical molecules. And also there are certain genetic molecules. You put the gene inside the plant for more growth and yield, it can do it. So both the ways you can do it. So which one you want to do? You want to use the chemicals or the biological? Biological, right? Environmental friendly.
Sir?
Yeah.
How can we make a normal seed into a hybrid seed?
Again, genetically you can make it. Low yielding and high yielding. Okay. If you take it, put them together, you might get a better one or average one. And the main thing is, for example, the high yielding one is not growing here in this environment, for example. Okay. And you are getting only low yielding one here. So you hybridize that one at the gene level, at the genetic material level, in this one which is growing here, then you will get the high yielding variety. That is how the hybridization is done.
Sir, there are two types of diabetes are there. What are there, sir?
Type 1 and type 2. Type 1 is genetic, type 2 is adaptive. If you don't exercise, don't eat good food, then you get type 2. That is in your hand. Good exercise.
Yeah. I think, sir, this is by far the most interactive session we had during the Milo CCMB scientist interaction, sir.
Oh, thank you.
Our students are very curious and they would be keeping you for another half an hour also if you keep interacting.
Yeah.
Thank you so much, students. And thank you so much, sir, for patiently answering the questions. I am
Thank you. I
sure you
really
also
enjoyed,
loved
but
interacting with our T-Series children.
yeah, as I told you, I really miss physical interaction with your students, but nevertheless, I really enjoyed. I'm really thankful to you and also for Somadata for giving me this opportunity. And I think Somadata has some questions to ask the students. So the small quiz, yeah, small quiz. So can I
Yes, sir. Yes,
hand
sir.
it over to
That's
her?
what. Yes, sir.
Thank you very much, Dr. GSN Reddy. That was, I think, a very, very interactive session, as Pavani just pointed out as well. And I hope you all had a lot of fun. And now let's do a fun quiz, just to summarize what all we have been discussing in the last 1.5 hours. So we will be doing this quiz on a platform called as Mentimeter. I am sharing the link in the chat box. So if you go, you will see the link in the chat box. I see quite a few of you joining in. Come in, write your name quickly. I'll wait for like 30 seconds and then we'll start the quiz.
Somdata, could you please share the screen of the quiz as well while playing so that we can all see how the students are participating?
Yes.
Yeah, thank you.
Come on, 38, 39 of you are here. I know there are more people. Quickly. Pavani, shall I go ahead?
Yeah, if you have 40 or so, you can start maybe.
Okay. All right, the first question is up. Which of the following species is the smallest? Virus, bacteria, fungi, plankton. Quickly answer. The faster you answer, the more points you get. And of course, it has to be a right answer. Cool, virus. Virus is the smallest. So 18 of you got it correct. So Shirisha was the fastest in this round. Let's see what happens next. Are all bacteria harmful to humans? Is it true or false?
False.
You don't say out the answers, you just click on your screen. Yo, it is false. We heard today how many different kinds of bacteria can be very, very useful to us. Nagavalli Prasad has now come to the first position. All right, let's move to the next question. Bacteria living in very hot temperatures are called what? Halophile, hydrophile, thermophile, or siderophile? Thermophile. Yeah, we discussed it quite a bit today. All right, Nagavalli Prasad has still retained his first position. Ooh. Probiotics are nutritional supplements added to processed food, food processing mechanisms, useful bacteria present in our food, harmful bacteria in our food. What are probiotics? It's the useful bacteria present in our food. And yes, they can also be sometimes added to other things, but basically those are useful bacteria. Nagavalli Prasad is still retaining his first position. Oh, Anusha has now come as the fastest person. Few bacteria survive unfavorable conditions by releasing special compounds, producing spores, moving away to favorable environment, or all of these. All of these. Some of them can release special compounds, some of them can produce spores, some of them know how to move away to other environments. So all of these are correct answers. All right, so here is our final scoreboard. So Anusha is the fastest. Congratulations, Anusha.
Congrats, Anusha. Who is Anusha? She a student or a teacher?
Yeah, teacher ma'am.
Hi, hello, Anusha. Ma'am. Yeah.
Where is Anusha? Oh, hello.
Yeah, hello ma'am.
So it's good that you understood everything that GSN Prasad was sir was explaining.
it's
You
very
can teach
interesting
the same
session,
to your
ma'am.
students who
Yes,
missed
ma'am.
the session today.
Yeah, excellent presentation, ma'am. Thank you for, yeah.
Great. So hopefully we'll meet someday soon. And until then, you all stay well and stay safe. And we'll meet again next month with a new talk with a new scientist. So stay tuned.
Thank you.
Bye. Thank you all for such lovely being such a lovely audience. Thanks.
Thank you all
Bye.
too. Bye.
Bye, Neil.
Thank you, ma'am.